Okay, so I think we're good to go. Hello, everybody. Let's get started. And thank you for joining the third Cancer.io Sebinar, Single Cell Tumor Immunity. And we're happy to host you today from all over the world. We're expecting today like maybe 100 to 200 participants. I'm your host today and chair, Heidi Haikala from Cancer.io. And Cancer.io is a Business Finland organized network for IO research and promoting the uptake of new immuno-oncology drugs. And it's coordinated by University of Helsinki. And a couple of practical things. The Sebinar will be recorded and it's going to be in our YouTube and web pages. So if you miss some part, you can always come back. We have first two excellent short talks and then followed by a keynote from Dr. Bernd Bodenmiller. And then we will have the brief scientific QA right after the three talks. So you can post your questions already to the QA function during the talks, but we will be addressing them after the speakers. And then from there, we will switch to the interesting panel discussion where we will be discussing the applicability of these single cell and spatial technologies in research and clinical decision making. And Jeroen Pauwels, who you maybe briefly saw, he will be moderating the QA chat and bring some interesting questions to me. And again, use the QA function, not the chat function. And you can always reach the Cancer.io office team, us, through the chat function or by emailing the Cancer.io office@helsinki.fi. And University of Helsinki students can get credits from this by signing up in WebAudi and sending us a short summary of what you have learned during the session and use our Cancer.io office email.
Quick note, WebAudi doesn't exist anymore. It's now something else, but
okay.
But the students probably know.
Yeah, yes, the students know better than me. So from there, we can move on to the first speaker. So we have Dr. Anniina Färkkilä, who is a MD and a principal investigator in the research program for systems oncology in University of Helsinki. And she just recently completed her postdoc fellowship in Dana-Farber and Harvard Medical School, where she focused on the mechanisms of resistance and biomarkers for combination therapies. And currently her laboratory studies tries to understand the complexity and cell-cell interactions in the tumor microenvironment using these highly multiplexed imaging and advanced bioinformatics technologies and functional models in ovarian cancer. And today, Anniina is going to give us a brief introduction to single cell technologies in immuno-oncology. And the floor is now yours, Anniina.
Thank you, Heidi. Thank you very much for this invitation. I will share my screen. So as you can see, so thank you very much for this, and I think it's a very timely topic and very interesting topic to me, and very happy to start this interesting session and seminar. And these are my disclosures. So when I thought about this topic, and one thing that I wanted to show and highlight is that cancer progression is actually an complex interplay of the cancer cells and the host. And to be able to understand cancer, we need to be able to understand the tumor microenvironment in detail. And these recent single cell technologies have really, we have started this journey into really understanding cell-to-cell basis, these different subpopulations of the tumor cells and immune cells, and also the stromal compartment, including the endothelial cells in the tumor microenvironment. With these single cell technologies, we can also learn a lot more about the phenotypes of individual cells. For example, here the tumor cells are not actually in just one state. So there can be multiple states existing inside one tumor, even within the tumor cell population. And these single cell technologies have recently also enabled us to trace individual cells or cellular evolutions using, for example, DNA sequencing, as shown here, where the different tumor cell clones show different dynamics across the time. So we can also trace the tumor evolution. Here is a brief overview of these constantly evolving technologies that we now have in single cell research. So as you can notice is that most of these technologies can be either antibody-based or sequence-based. And many of these technologies, as shown here, they are, for example, cytometry, flow cytometry or mass cytometry, sequencing technologies. They require either blood as a source or dissociated cells from the tumor. Recently, there has also been these technologies emerging that don't actually require dissociation of the tumor cells, and they can analyze single cells from these fixed tumor specimens. And these include spatial transcriptomics, imaging mass cytometry, which is where the detection is antibody-based, but it's detected by mass spectrometry, and multiplexed immunofluorescence, which is, for example, completely antibody-based and immunofluorescence detected. These new technologies, of course, they bring unprecedented depth and quantity of the data, but they also bring a huge challenge to the data analysis. So the depth and the amount of data is really huge, and they challenge also the infrastructures and the bioinformatic pipelines. And recently, there's been, over the recent years, there's been a recent very vast developments in these data analysis platforms and pipelines, where we can actually try to learn and understand more of the single cell phenotypes, also in the cellular context, and hopefully then link these to the patient outcomes. If I think about the clinical context, where the understanding of the tumor microenvironment is really critical, is the context of immunotherapies. So these PD-1, PD-L1 blockade, basic immunocheckpoint blockades are really entering the clinical use in many tumor types. And at the moment, there are 6 immunotherapies approved by the FDA, and they are in clinical use. This is a timeline of these FDA approvals from 2014 to 2020, and you can see the really kind of emerging FDA approval rate. These are the 6 drugs, and these are the different tumor types. However, what is not notable here that there is, in the vast majority, there's no biomarker existing for these cancers and these treatments. And at the moment, there is still no consensus on whether these biomarkers should be used and what would be the biomarker. With these new technologies, there was one recent pretty interesting meta-analysis comparing different modalities of biomarkers in this clinical context. And they did show that learning more and seeing more at the cellular resolution, these multiplexed technologies can really be superior to simple conventional immunohistochemistry, also in predicting responses to these therapies. And there are some spatial features that were shown in one study that that could be linked to improved responses, and they could serve as biomarkers. So in my work, I have been working in this field, and we set out to search for these types of biomarkers in a combination trial, which combined PARP inhibitor drug and immunotherapy PD-1 drug in ovarian cancer. There were 62 patients in this trial, and the objective response rate was 18%, which to me means that only 18% of the patients really responded, and we need better biomarkers to identify these patients. And here is the waterfall plot where you can see that there were even two patients with complete response where none of these conventional biomarkers were able to identify. So first we did kind of traditional genomic analysis. So we find a mutational signature tree, a mutational signature that was associated with response. And we also find in gene expression profiling that these interferon signals and exhausted CD8 T cells were associated with response. To understand more about the mechanisms and biology in these tumors, who responded and who didn't, we utilized this single cell profiling using multiplexed immunofluorescence. So this is a technology that is used on formalin fixed paraffin embedded slides. And in this study, we used 42 biomarkers for cell types and functional states. And after the cyclic staining, the images are processed and the data is quantified at single cell level. And then we can analyze the data using bioinformatics in spatial analysis. And in this study, we first annotated the 7.2 million single cells into the tumor, stroma, and immune. And then we were still further able to subclassify the immune cells into these deeper subpopulations. And what we found is that the signals that we captured from the nanostring data, expression data, actually were coming from the same subpopulation. So the exhausted CD8 T cells had a higher interferon signaling in the responders. And this is the same verification in the tissues itself. And we next wanted to learn more about whether these, how does this mutational profile and these exhausted CD8 T cells communicate in the tumor microenvironment, and we performed neighborhood analysis. And we asked, what are the PD-L1 positive neighbors that are surrounding these exhausted CD8 T cells in the responders? And our answer was the tumor cells. So we found that the PD-L1 positive neighbors were more often tumor cells in the responders. And this was especially true in these tumors that had this mutational signature. So we found these spatial features of these tumor cell and immune cell subpopulations that really were enriched in the responders. And these are the commonalities that we found as biomarkers in the responders. So there was a really crosstalk and also enhanced spatial signaling of the signatory positive higher PD-L1 expression tumor cells, macrophages, and exhausted CD8 T cells with interferon signaling. And this, you can read more about this story in the paper that was published last year. So here I am concluding and giving the floor to the next speaker and acknowledging Julia Casado, who was presenting, performed most of the analysis, Peter Sorger, who has developed this multiplex technology at Harvard, where I was doing my postdoc, and also Alan DeAndrea and Panos Konstantinopoulos for leading the clinical trial. And looking forward to the rest of the seminar and the question.
Okay, thank you, Anniina. I'm happy you pointed out the complexity of these algorithms and softwares and everything, because that's something we want to discuss more about in the panel discussion that is upcoming. But thank you, Anniina. So we're moving on, and if you are having any questions, you can post them right away to the QA, and we will address them after the three talks. And next up is Päivi Saavalainen from SCellex, and SCellex is a startup company operating in the University of Helsinki, and they are developing spatial transcriptomics and chip platforms to study the gene expression profiles of cells directly, either from tissue sections or single cells. So Päivi is going to tell you more about this.
Yes, thank you. I have troubleshooting now between two screens to have this I have had some troubleshooting this now quite a lot, but my laptop doesn't want to. Do you? Which one do you? Because I don't see now.
We can see your full screen with the PowerPoint, so you
okay.
Can, in the worst case, you can also go with this one.
Yeah, I think because my laptop almost crashed when I tried to switch the.
Okay, so maybe this is smarter to go with this.
Yeah. Let's go this. Okay. But now I'm actually staring a little bit different camera, but to see my slides. Oh. But sorry for this. But anyway, so I will just give a very brief overview of the spatial solutions for single cell omics. Yeah, I'm university researcher in the Helsinki University and Färkkilä Research Center, but also running the startup company SCellex here in the same campus. And so single cell. Now I'm talking about transcriptomics and DNA and RNA based methods, because in the Anniina was mentioning more also the proteomics level and the antibody focused levels, but I'm kind of here comparing the transcriptomics methods. So what we can, this is really annoying now to get this working.
Now it works.
Better?
Yes,
okay,
now it works.
Great,
excellent.
Great. Yes. So as you know, the suspension cells, the single cell resolution in suspension cells, it has been revolutionary for many years. But you need to kind of dissociate the tissue to run the single cells in suspension to droplets, just like in 10x genomics. Chromium is the mainstream method for that, and that of course needs enzymatic and mechanical treatment of the solid tissues, and it doesn't give you the spatial information. Then another traditional way to do this is a laser microdissection to cut the single cells one by one from the tissue sections and sequence that, but that is not very high throughput, and it's very, very slow and expensive per cell. Then there are the in situ hybridization and probe-based methods to kind of visualize each RNA by fluorescent probes. For example, nanostring methods, which are great and high throughput, but and can be multiplexed a lot for hundreds or thousands of genes in multiplex method. But what is important maybe in cancer and especially in immunology is the sequence-based information, because you need to maybe to detect some mutations in sequence level or to find novel gene markers and not just previously known gene sets available for the probe assays. And then also in the immunology, you really might want to look at the clonality of the T cells and B cells. I mean, what are the T cell receptor sequences and clones within the tumor tissues or other immunological questions, and also the B cell heavy chain and light chain sequences if you look at the clones. So the clonal level information also requires the actual sequencing of the RNAs or DNAs. And for that, the mainstream at the moment and the only commercially available system is from 10x genomics, the Visium platform, where you can really use that as a microarray type of spots, 5,000 spots per area, and you put the tissue on top of the probes and capture the RNA, and you then get an overview of all the genes that are expressed in those spots. The problem has been now, but it's not single cell resolution. The spot size has been 55 micrometers, so it gives an average of several cells within each spot area. But the Visium, the kind of higher resolution, I think 10 micrometer version is to be launched soon by 10x, so the resolution will be better there. What we are developing in our, well, this is just an example of 10x Visium platform, just from a paper from others, just showing very nicely the power of the Visium methodology and the tissue transcriptomic image there, to showing that the myeloid cells and the cytotoxic T cells co-locate in the same part of the tumor, and the interferon expression pathways are up there as well. So it's kind of nicely demonstrates the power in cancer immunology as well. What we are working on, this is not yet in the market, but we are developing another method for single cell capture or tissue direct capture from the tissue in SCellex, and we don't have a microarray barcodes on the slide. Instead, we have a patent pending barcoding method for pico wells, and it's quite flexible with the size. At the moment, our prototype has 15 micrometer diameter wells. We can put, if we fill the whole microscope slide with it, it will be several millions of these, but of course you don't need the whole slide area for a tiny biopsy. But like 1600 wells per squared millimeter biopsy, it's the current resolution. So we barcode those wells, and then your cell, the tissue sample, your cryosection can be on just a normal microscope slide. You can fix and stain that, or plenty of them, store them in just normal glass slides, and then later select that which ones you want and which part of your tissue you want to do spatially. Because then our barcoding is in our special chip, and then you put them together and a lysis buffer between, and you capture the RNA to the wells. And this gives also flexibility of the sample type. So we are currently trying to optimize this also for blood smears or other cell smears or cytospins of suspension cells, so that you could archive the cells and samples, fix them, stain them, and archive them in freezer, and later on do that also for fixed cell collections. And what is another interesting feature is that we can utilize these pico wells directly from the cell cultures. So we could, this is also something that we have just started to optimize, but we could do high throughput single cell cultures in our wells, and add there some labeling agents, for example, some interesting markers or antigen-specific cells, label the antibodies that they secrete, or do CRISPR screens or drug screens, or detect some secreted molecules out of the cells. Take the image, the spatial image, and then sequence the cells and in all of the cells, and then link later the sequence data and the transcriptomic data to the actual well picture to see that which did it come from the well that had this marker signal up. And this of course creates a huge amount of data, layered data, X, Y, Z layered data for every gene, and for this we are utilizing machine learning methods to link really the well coordinates and the sequence data and the original microscope image of the cells or tissues together to software. So this is, we have lots of things to do and optimize, but if we compare our method to the other ones in the market, then especially the high throughput and pricing per cell will be very, very cost efficient, and it allows also the sample archiving and functional assays, as I explained. So this is our method will complete kind of complete the other existing methods to have more options for the users. So this is just a short introduction to what methods there are available, but this is our small team expanding at the moment, and I'm looking forward to get our method ready so that we could start doing pilots with the local researchers first, and then hopefully go to market at some day. Thank you.
Thank you, Päivi. Really exciting technological developments, and I'm sure you can add some also perspective to the panel that how can these kind of innovations be taken from the academic bench to the actual market eventually, hopefully. So that's going to be later on. And if you have any questions for either Anniina or Päivi, you can send them using the QA function still. So we'll be addressing them later. And now we move on in the program. So we're heading to the keynote speaker, Dr. Bernd Bodenmiller. And he is a dual professor at the Department of Quantitative Biomedicine in University of Zurich, and also in the Institute of Molecular Health Sciences in ETH Zurich. And his research investigates the mechanisms of cancer ecosystems, and his work has already identified novel tumor and immune cell phenotypes associated with clinical features, and also signaling mechanisms leading to therapy resistance. Also, these methods that he has been developing already have already widely been used by researchers, but also in clinical studies. So we're actually really interested to hear what you can bring to this single cell spatial discussion topic, and really excited to hear about the latest developments from your side.
So Heidi, really thanks for the nice introduction. And also thanks for giving me the opportunity to present in your webinar or Sebinar. You still have to explain to be the B instead of seminar, but a Sebinar. But in any case, I just want to warn you, I have a cold, a non-COVID cold, and once in a while I'm coughing. So apologies for that. Okay. I assume you can see now my cover slide.
Yes, looking good.
Perfect, thanks. So in my group, we are working on developing novel experimental and computational methods for the multiscale analysis of tumor ecosystems with the aim to ultimately impact and improve patient treatment and care. So as you know, in classical pathology, a pathologist looks at the tissue morphology and at one or few markers. And while it's amazing what a pathologist can tell you just looking at the tissue using an H&E or IHC stain, they will all agree that the information they can retrieve from a molecular point of view is very limited. And this is one of the reasons why current pathology does not enable personalized treatment suggestions. But we have those standard treatments, the ESMO guidelines, and based off those, patient groups are formed for treatment. Now, what we try to achieve is to perform in situ tumor biology measurements of every single tumor so that we know the cause of the disease, and based on that, bring the right drug to the right person by matching the treatment with the molecular cause of the tumor disease. And this then automatically begs the question, which features of a tumor are actually informative? Now, you all know by now that tumors are not just a mass of tumor cells, but that there are different tumor clones. The tumor is embedded in the stroma. We have immune cells, stromal cells, nerve cells, blood cells, etc. They're really embedded in a very complex environment. And so ultimately, what we would like to know about a tumor is what cells are present, what is the composition of the tumor, how do cells interact, so where are they located, which receptor ligand interactions do they communicate, molecules they release. Of every cell, we would like to know what are the deregulated pathways. We want to know the overall 3D morphology. And ultimately, that means that we need to make enable measurements from the level of molecules to the level of pathway cells, all the way to the 3D morphology, meaning we need molecular measurements with spatial resolution. So all of these, what I call the scales of a tumor ecosystem. Why do we want to measure all of them? Simply because all of them are useful for suggesting treatments. If we know a special tumor cell phenotype, we can target it. Anti-HER2 treatment is such an example. We can target deregulated pathways. Targeted inhibition of the MAP kinase pathway in melanoma is a beautiful example. We can target the 3D morphology, and anti-angiogenesis drugs, again, are a good example of that. We can also target cell-to-cell communication as exemplified by the PD-1, PD-L1 inhibition, but interaction that we then inhibit, reactivating in many patients now the immune environment. And in addition to being useful for deciding on personalized treatments, such measurements are also highly informative just to understand better tumor biology, and ultimately also to stratify patients that then reflects this precision treatments. Now, the technology that we have initially developed to perform this highly multiplexed molecularly and spatially resolved measurements is called imaging mass cytometry. In imaging mass cytometry, we take a routine tissue section from a pathologist, we then stain it with antibodies that carry pure metal isotopes that were put on the antibodies. Then once those antibodies have bound to the antigen, we take a laser that systematically rasters over the tissue. And every time the laser hits the tissue, it generates a small particle cloud. And this particle cloud is then transferred into what is called the mass cytometer, or in short, CyTOF. And here, the tissue is atomized, the atoms are ionized. And in the end of every individual pixel, we read out which antibodies, we read out which isotopes are present in the location where the laser shot. And by inference, we know which antibodies are present in a given pixel location, as well as their abundance. And ultimately, every laser shot corresponds to 1 pixel in such a raster image. Okay, so by now, these imaging mass cytometry images, they're really akin to fluorescence microscopy images. Here you're looking at a cell line called A431 cells. And the main thing I want to point out is to the lower left. If you look at phosphorylation on histone H3, which indicates mitotic cells, you can actually beautifully see how the chromosomes of the to-be daughter cells are lined up and how they are pulled apart and being separated. And that's really something you can also see with a routine fluorescent microscope, and we really now see the same things. That's a different cell line, MDA-MB231. It's a breast cancer cell line. In green, you see vimentin, in blue, DNA, and in red, CD44. Again, if we zoom in, you can see that we can measure here the vimentin filaments of individual cells, or here again, a cell in late mitosis. And we did really lots and lots and lots of validation comparing imaging mass cytometry to fluorescent microscopy. This was done here, staining with fluorescently and metal labeled antibodies the same tissue. And you can see that it is pretty much the same. We also compared it to IHC. And again, we can see that we can nicely recapitulate what IHC tells you, but with the important differences that we can measure in the moment over 40 markers simultaneously, and that we do not suffer all of the artifacts you have to face in fluorescence microscopy, but also in IHC techniques. And currently, we routinely measure over 40 markers. It takes us 1 hour to measure a square millimeter, so it's still rather slow. The limit of detection is similar to fluorescent microscopy. We can do 3D tissue screening, and the approach was licensed and is already commercially available since roughly a couple of years. So there are several advantages over fluorescence-based approaches that we have with imaging mass cytometry. And to illustrate you that, I show you to the left it's a breast cancer bone metastasis section. May you see in yellow pancytokeratin, in red CD68, and in blue you can see DNA. And if we now here go and zoom in, you can see the same region over here that was highlighted here in white. You can see it here highlighted to the right with the same colors, same markers. And the very same region now here imaged with imaging mass cytometry.
And there are several things that you can immediately see. So first, here to the upper right and to the lower right, there's lots of signal on the cytokeratin channel. And we do not see that in imaging mass cytometry. There's also lots of yellow background signal. Again, we don't see this in imaging mass cytometry. And here you're looking at autofluorescence of this tissue. We do not have this. And you also see that we have a much higher, this also shows you that we have a much higher dynamic range in imaging mass cytometry compared to fluorescence microscopy. We have about 1,000-fold higher dynamic range. Then also compared to serial fluorescent imaging methods, we do not have any tiling effects that you often see, and we really do not have to optimize the antibody order, because in the end, we stain the whole tissue with the antibody mix simultaneously. Here are also a few example images from an observational clinical study from a melanoma tumor. You can see here in red, these are cytotoxic T cells. You can then see here to the left that they express PD-1. To the right, you see in blue the tumor cells. You can see over here that these actually express PD-L1. They also in blue express MHC class 1. And there were many more markers that we looked at. And all together, this then made us, convinced us that this patient should get immune checkpoint inhibitor therapy. So before I mentioned that our technology is not the fastest and it takes about an hour to analyze a square millimeter. So what do we do about big tissue sections? Well, it's very simple. We stain the tissues with the metal antibody mix, but we also throw in fluorescently labeled antibodies that indicate to us regions of interest. So here, for example, we were interested in the islets of the pancreas. This is a pancreatic section, probably like, what is that, 3 × 2 centimeters. And then we identified, we scanned this then overnight, the whole tissue section. And then based on the fluorescent-based signals, we choose regions that we image with imaging mass cytometry. And this is a really easy approach since you can, I think, scan 100 slides per night in the slide scanner. And so it's very easy to identify regions of interest with high throughput. We then also develop technologies not only to measure proteins and their modifications, such as phosphorylation sites with antibodies, but we also wanted to be able to measure transcripts. And the reason is that many of the really interesting molecules, such as cytokines, chemokines, etc., they are secreted first, so you don't see them in a cell. They're very diluted second, and third, they're not a lot of good antibodies. So then what we adopted is a branch DNA approach where you build like a DNA Christmas tree on a transcript. Then we can stain with the metal labeled antibodies, and we can come in with images that bind to the DNA that carry metal isotopes. And in the end, you can simultaneously image, this is again a breast cancer tissue section. We can simultaneously image the tumor cells shown here in blue, CD16, CD8, so here the T cells in green, but then CCL18, CCL5, CXCL9, these were measured on the transcript level, shown in light blue, pink, and yellow. And down here are a few other ones that we also measured in this tissue. And by now we can routinely measure 12 transcripts together with 28 antibodies. And the manuscript describing this, you can actually find on bioRxiv. So initially I also told you, we want to be able to see tissues in 3D. And to enable this, we developed a workflow where we take a tumor block, we do serial sectioning of the tumor. Each section is 2 micrometers thick. And we collect all of these sections. We go through the routine imaging mass cytometry workflow. We then register, we then do the imaging. We then register each image computationally. That provides a 3D voxel model. We can do single cell segmentation and continue with the, for us, routine data analysis. Here's now an example of a HER2 positive breast tumor. That's the voxel model. You see in yellow, that's the pancytokeratin. Smooth muscle actin is shown in pink. In red, you see blood vessels, and you can see in white the immune cells. Then after single cell segmentation, and that is shown here, we can really delineate the individual tumor cells, shown in orange. And then here in the stroma, we have cells high in vimentin and smooth muscle actin. So we have both immune and stromal cells, such as fibroblasts, surrounding the tumor mass. And you can see to the right, also being within the tumor. Now, why 3D? Many people ask me, why do we ever want to know how a tumor looks in 3D? Good question. Well, first of all, we are 3D objects. So probably it's good to analyze us in 3D and our diseases. But to give you a more specific example, so here you see in yellow the surface of the tumor mass, and the rest is in the stromal region. Now, please look here at the center. Now we have here this micro protrusion and many individual tumor cells. Let's wait for a second that it repeats. Again, here we have the micro protrusion and now many individual tumor cells. And so what we are looking at most likely is that cells become metastatic. And that was pure chance that we hit such a region in this tumor model. And now what we can do in 3D, we can look at this pseudo-spatial temporal process and actually see how the phenotype of those cells are changing from being in the tumor mass to this micro protrusion to becoming individual and potentially invasive cells. And just with one 2D section, you would never be able to see something like this or investigate it the same way as we are doing that now in the moment. Finally, it was also said before, such technologies generate a lot of data. True. And for that purpose, we develop tools, computational tools. One is called HistoCAD, where you can segment, you can look at the cell phenotype clusters that are present. I should actually show it on the images. You can plot also which phenotype is present on a tissue. You can determine cellular neighborhoods in a statistically sound manner. We can also look at regions. And you will see later on that we can also look at distances, communities of cells. So we can derive really lots and lots of different features from these tissue images. And to make this more broadly available, we will soon publish the so-called HistoCAD web. There's already the HistoCAD available, but soon we have a cloud-based version. And then there's also a beautiful R package called Cytomapper that allows you to do such analyses in R. And you can find all of these tools on the Bodenmiller Group GitHub page. All right. So just quickly to reiterate the current status of imaging mass cytometry. We have really a high dynamic range of up to 10 to the power 5. We can analyze any tissue as long as you get antibodies on it. The limit of detection is like fluorescence microscopy. It takes about an hour to measure square millimeter. The resolution is a bit lower than fluorescence microscopy, but you saw it doesn't really make a difference. We can measure transcripts, proteins, and modifications. We can do the whole slide analysis. We have very few artifacts. I want to stress this. We can do imaging mass cytometry now in 3D. What I did not show you, we developed a high throughput screening approach for 3D tissues, such as tumor spheres and organoids. And really, the technology by now, and I will show you later on more, is extremely robust and reproducible. And we can highly reproducibly measure 40 markers. And because it's now so robust and reproducible, typical projects in my group are in the range of thousands of patient samples. And this, I believe, is also really necessary to make robust and statistically sound observations in a disease that's as heterogeneous as cancer. At least you need hundreds of samples, but obviously the more samples you get, the better your statistics get. All right. And since now imaging mass cytometry is such a solid technology, it's already used in many international consortia. So we are part of the Grand Challenge UK IMEX team, where we study tumors in 3D. We are part of the EmuCam project. That's an IMI European funded project together with Merck. Then we are also part of the HubMap project, where we look at the immune organs. And in the end, I will show you a few slides on our ongoing observational clinical trial that is called the Tumor Profiler. So now that I've shown you the technology and I told you, well, it's so amazing to have this data, we really still had to answer, can this multiplex tissue imaging technology that can define so many different cell phenotypes, will it actually be helpful in any clinical question or setting? And so what we wanted to ask is, can the single cell pathology actually better stratify breast cancer patients? To address this, we first determined the composition of the tumor ecosystems in breast cancer. We evaluated the structure, spatial structure within this tumor ecosystem. And then we assessed which of these ecosystem features actually can stratify the patients. And so for this, we looked at the initial cohort, and then there was another validation cohort. But we had an initial cohort of 281 cancer patients, for which we had long-term survival data. So we really knew of every patient, whether or not she died of breast cancer. And now, what did we want to achieve? To the right, you see the survival curves of the main breast cancer subtypes in red HER2, luminal B in violet, in green triple negative, and in yellow, a luminal A breast cancer. And please note, nowadays, those curves look somewhat different. The order is different due to new therapies. But what is still the same is that at a given time, let's say 10 years after diagnosis, a fraction of patients will have died of the disease, no matter which subtype, and another fraction luckily still alive. And the question really is, can we actually separate those clinically molecular subtypes into more finely grained subgroups of patients, like those that have a great long-term survival versus those then that have a poor long-term survival? To address this, we generated an antibody panel that contains many of the lineage markers for breast cancer, the standard markers for pathology like ER, HER2, Ki67. We had the immune context, mesenchymal markers, endothelial markers, transcription factors, cell growth markers, epigenetic mark, cell death marker, one for hypoxia, etc. And in the end, we analyzed about 1 million cells in this study. We then first determined which single cell phenotype clusters, meaning groups of cells that have a similar phenotype, exist, shown on this heat map here. On the x-axis, you see the markers we measured. On the y-axis, you can then see the different clusters, and the color scale indicates from blue lowest to red highest the expression of a given marker. Now, to simplify our downstream analysis, we took markers that biologically were very similar, and we derived meta-clusters. And then here to the right, you see the different meta-clusters. So in green, we have an immune meta-cluster. In light blue, immune meta-clusters, plural. We have here the stromal meta-clusters, and the different colors down here, they're all the different tumor cell phenotype meta-clusters. And then we first looked at the molecular subtypes used in the clinics. So here we have the luminal A, luminal B, HER2 positive and triple negative patients. And this was a classification given to us from the pathologist. And then we plotted for these 4 different molecular subtypes which immune, stromal, and tumor cell phenotypes are present. You can see that some of these phenotypes are present in a higher frequency than others, and they also roughly correspond to the molecular markers that are used in the clinics. But for all of them, you see actually a wild mix of also other tumor cell phenotypes. And it simply tells us that the current classification scheme does not reflect and take into account the phenotypic richness that is present in these tumors. So we then turned our analysis around, and we plotted for every patient the density of the tumor cells and of the different immune and stromal cells. Then we performed hierarchical clustering across all of the 281 patients. And now just looking at that, you can immediately see, whoa, there are groups just based on the color of the tumor cell phenotypes. In total, actually 18. And then came the big moment for us. So now we had this single cell phenotype, and we call this the single cell pathology groups defined. The question is, now if we look at the survival curves of those phenotypically defined breast cancer subgroups, do we see randomness and randomness in survival? And for our cohort, again, the survival curves are shown here. And in black is the average of the whole cohort. And so if these single cell pathology groups, if they would be random in the composition of women and in their survival, then we would be very close, or we should be actually at the black curve. And the other way around, you can see here the triple negatives that the worst prognosis, luminal A pretty good, HER2 the best in this group. And here we have the luminal B, they have like the second worst prognosis. Now the question was, if we now just look at this luminal A tumors, the orange one, what do we see for the single cell pathology groups? And you can see that we could identify a subgroup of patients where none of the patients succumbed to the disease, and other now mostly luminal A patients that had a poorer than expected survival. So we could split up this molecular subgroup used in the clinics into a long-term survival group and those that actually do worse. And we also did this for the luminal Bs. And also here we see that we can differentiate the long-term survivors versus the others. And even for the triple negative patients, we could identify those patients that had an excellent long-term survival versus those that then had a poorer survival. So this showed us that highly multiplexed imaging really can nicely stratify patients with long-term survival. It also showed us that this technology and approach holds a lot of information that could be useful in the clinics. Another thing that is really fantastic about this type of data is you have images of cells with markers that you can interpret. And so if you now have questions about the survival curves, you can go back to the images and start thinking about it. And one thing that we actually found looking at this survival group number 1 and the group number 3, where we have less survival, the tumor cell phenotype present was the same, but we saw that the overall spatial structure was different, and this number 3 also had more of this pink phenotype. But really, the spatial structure struck us. And therefore, we then thought, how can we computationally describe that there are tumors where we have more cells together and other tumors where cells are more dispersed? And therefore, what my student Jana Fischer thought, well, we could simply determine whether there are cellular communities in the tissue. And in order to generate those communities, or not to generate, to determine those, we determined the cell phenotypes. We made a graph where neighboring cells are connected by an edge, and each cell is a node. And then we used the Louvain algorithm that also Facebook uses to tell you who are apparently your best friends. And we used the same algorithm to figure out which are the best friends of a given cell. So to whom is the cell mostly connected with? And this then gave us those cellular communities. So then when we first looked at the tumor communities, we actually were quite struck. Because if you look here at their size per community, here are the different communities on the y-axis, you can see that pretty much every community consists of a single cell phenotype. But where they differ is actually in the size. And so here we really see that these tumor communities are driven by homogeneous phenotypes and the area. And what I don't show here, but what we see now over and over again, even if a tumor is highly heterogeneous in the tumor cell phenotypes, typically those phenotypes form pure communities, but they do not mix up with each other. And when you look at the strobal communities, we see that there's a mixture of cells. So these are driven by the composition, but you can also see that they're driven by the size. Now, when you go back into the single cell pathology group 1 and 3, you can see that this one is driven by the tumor community 23, and the one single cell pathology group 3 is driven by the community 17, describing the different sizes of the same tumor cell phenotype. Ultimately, we then calculated the hazard ratio, and we plotted the hazard ratio for the different communities and for the molecular subtypes. And to the left, you have the poor prognosis. To the right, you have the good prognosis. Now you have seen that those patients with this tumor community 17, so small clusters, they clearly now, indeed, we see again have a poor prognosis, while the ones with the sheets have a good prognosis. Then the other way around, once we knew that, we systematically used the community analysis, and we found even more extreme examples. Like here, where the same phenotype, but being in a differently sized communities, has quite divergent prognosis for the patients. And we saw this actually over and over and over again. What was really also interesting is that we had these stromal communities associated with good prognosis, and they were typically found among those triple negative breast cancer patients that had this excellent survival. Okay, so now I've shown you that this highly multiplex imaging of patient samples holds clinically useful information. But then we also wanted to know, is imaging mass cytometry actually clinically useful? Meaning if it's used in an actual clinical study. And for this, we started in Zurich what is called the Tumor Profiler Project. It's an observational clinical study where we took patient samples, we profiled them comprehensively, as you will see in a second, and then we do data analysis for the benefit of patients. And the core question was really, are those technologies that we included here, can they be used in the clinics, and are the data useful? And so what we did, we took patient samples from a given patient, we took biopsies. These went then through the routine diagnostics in Zurich, which is IHC, digital pathology, and gene panel sequencing. And this then provides a standard clinical report. But then in parallel, we took from the same patient also tumor samples, and we performed single cell transcriptomics, single cell genomics. We did single cell proteomics using mass cytometry. We did single cell tissue analysis, that was imaging mass cytometry. We also did drug response testing, etc. And all of this then also went into a summary report that went to the tumor board. And in total, we have by now analyzed 240 patients, mainly melanoma patients, but also ovarian cancer patients and AML samples. And the workflow that we developed here in Zurich is that we collect those samples, then we integrate the data, we generate a molecular research report. This research report is discussed by scientists and clinicians in a pre-tumor board. And this pre-tumor board makes treatment recommendations only based on the standard guidelines and the standard data that's used. Then treatment recommendations when the additional emerging diagnostics is included, such as next generation sequencing. And third, if also all of the tumor profiler data is included. And in this summary report actually goes to the tumor board, and the tumor board then takes the actual treatment decision for a given patient. And so in my group, our role was we were the central lab, so we processed all of the samples, but then most importantly, we took the samples and we analyzed them by site-off, so suspension mass cytometry, and also by imaging mass cytometry. We did the data analysis, and then we uploaded the report to the hands of the pre-tumor board. Now, several things we really learned are key to have such technologies be used in the clinics. First, the data really has to be highly quantitative, because in the end, you want to automate the whole data analysis pipeline. And you know the sentence, garbage in, garbage out. That's even more true in this setting, and garbage out is even dangerous. Therefore, having data such as with site-off or imaging mass cytometry that has basically no artifacts was incredibly valuable to automate this whole workflow. Then we also had to implement the 2-week turnaround time. As I will show you, the analyses were highly robust and reproducible. Our analyses were pretty much failure-free. And we could also show that they can be integrated, the data, into clinical decision-making. And so this IMC workflow, by now, within 2 days even, we can send the report to the hands of the oncologist. And we achieve this by including references that are always ready to go, antibody mixes that are always ready to go, that can be stored long-term. The acquisition automated, data analysis is automated, summary generation is automated, to the point that everything is uploaded. And the key aspect here is that using imaging mass cytometry only requires standard FFPE tissue sections on a glass slide that are produced in the dozens of every single patient for the for the pathologist. The pathologist just takes one of his slides and hands it over to us, and we can generate in our report. And the technology is actually also quite high throughput. Based on all of this data, we generate an automated report that reports on cell composition, on the cell phenotype and state, cell-to-cell interactions. And since we built our antibody panels to inform on drug treatments, in the end, we can do drug predictions. And I would like to underline that of the 240 patients that we have analyzed, in more than 99% of the cases, we successfully generated a report. Now, I just also want to give you a quick example on how or why such new technologies can be useful. So here we have a stage 3 melanoma patient, untreated, and we analyzed the cutaneous metastasis. Then pathology couldn't really say whether or not this patient should get immunotherapy because it wasn't a clear-cut hot and cold case and expression of PD-L1, etc. So all of this wasn't too clear. Genomics was borderline with the tumor mutational burden, which in itself is not a great predictor. So that was borderline. And pathology and genomics did not inform in any way on targeted therapies. So then when we looked into our data that we have generated, it was clear-cut that the T cells were exhausted at high levels of PD-1, interacted with PD-L1 expressing tumor cells, and the tumor had still expression of a fairly high level of MHC class 1. For us, it was a clear-cut case for anti-PD-1 treatment. And then we also, looking at all of the signaling in our data, pathways that we measure, we also saw that there's a strong indication for sorafenib treatment and an anti-CDK4/6 inhibitor. And until today, actually, the patient has responded to the treatment and did not relapse yet. And with this, I want to conclude that imaging mass cytometry can also be used in the clinical context. Now we are really working on pushing the technology to the point that it can be more routinely be used in the clinics. And I greatly want to thank my whole lab. Admittedly, it's not the most up-to-date picture. It's a pre-corona picture, but we are soon planning to have a new picture. And I really want to thank everyone in my lab for their amazing work. Then I want to thank the University Hospitals of Zurich and Basel. They're both part in the Tumor Profiler project, as well as ETH Zurich and Roche. And I also want to greatly thank PATH. It's a foundation in Munich that provides us with an incredible amount of valuable patient samples. And then I also want to acknowledge the Swiss National Science Foundation, European Research Council, NIH, the HubMap Consortium, Cancer Research UK for the funding of the research in my group. And then I think later in the panel discussion, you can also ask questions about my research. Thank you.
Thank you, Dr. Bodenmiller. And I absolutely want to congratulate you for coming up with these great methods for the community and for the clinical trials as well. That this is really sophisticated way of analyzing the patient tumors. And also, I was impressed about the 3D resolution that you were showing in the images at some point. So it seems that every time I see a new talk from this topic, it's really like moving on with such speed that it's really, really overwhelming. there was a couple of questions from the audience, and actually maybe I can start with my own. So since there's such a huge amount of data coming from all of these different approaches, then what is the role, if any, for artificial intelligence here? Because also that field is developing really fastly and probably aligns really well with all the data processing.
I mean, so if we talk about artificial intelligence, then we typically talk about deep learning. And I think in the future, there's really a big value in using it. But in order to train the CN, so the convoluted neural networks, you need a lot of data. So we have one project in the lab where we have a cohort where we know the genomic status. And so we actually wanted to learn whether based on the multiplex imaging data, we can predict genomic features. And it somewhat does work, but the problem we're running in is that like having 1,000 images is simply not enough. You need huge data sets, and then you also need a very clear task for which you can train so that in the end you can really use the neural network. But you made a really good point. These technologies become more and more commonplace. The data sets will become bigger and bigger and bigger. I'm also sure there will be more and more international consortia that under controlled condition will generate gigantic data sets. And yes, then it will be absolutely valuable to have artificial intelligence approaches.
Excellent. Some of the panelists were saying they cannot ask the questions in the chat or in the QA, so you can just post them in the chat if you want to, or then just show your face and ask the question live. Couple of more questions from the audience. So there was a question saying that processing of the ICM takes 1 hour per square millimeter. And is this a limitation for pathology? And is there something to be done to speed this up? I think this was referring for the one of the first points.
Yes. So clearly the technology will become faster. That is foreseeable. It's a technology. Technologies become better. It will become faster. What I have to say, though, is that the throughput that we have in the moment is already equal or higher to what the gene sequencer does in genomic analysis of patients. So, and I think the data we generate is much closer to a gene panel sequencing analysis than to a standard IHC, because we measure 40 plus antibodies, but what we extract from these images is so much more than just individual markers. And so I think it's better compared to genomics, and then we already have a higher throughput than the genomics lab per machine. In fact, also the cost per sample probably is now similar or a bit lower than that. So we are comparable, but in the future it will be quite a bit faster. So that is no question. And still what we also do in our studies is that pathologists, so they are very important. Pathologists point out to us which regions we should image on a tissue. Because, pathologists, they say, oh, that's just necrosis, there's just this. That's really the region we should look at. And that's then where we zoom in with our technology. So that's really a mutual benefit of bringing those two together.
Right, and it's nice that you mentioned the cost as well, that it's coming down, because that's something I also wanted to ask about. But maybe I'll give the floor to Anniina again. She is raising her hand to ask a question.
Yeah, thank you very much, Bernd. It was very, very exciting. And as a clinician, it's very nice to see how these technologies really, come closer to the clinics for the actual patients. I wanted to, first, congratulate on the really, the fast pipeline. For example, in ovarian cancer, we don't typically need those results after the surgery so soon. But if you can do it in 72 hours, that's really like a Formula One of biomarkers. But I wanted to ask, how did you select the targets for the panel for the antibodies in the panel for these targeted treatments? And especially considering ovarian cancer, and what do you compare those with? Like, how do you assign that this tumor has a high CDK4/6 signaling?
Yes. So, there are two questions that you ask. So The first one is we took the drug information packages of the, how many are there? 56 drugs that are really commonly used in oncology. We took those and then we worked backwards and asked, what do we have to measure that might inform on a molecular level about those drugs? And so we developed an antibody panel that reflects results of pathways or processes in tumor cells that indicates that a given drug might actually be used, or like already known standard biomarkers.
Is that panel available somewhere?
No, not yet. And so that was one approach that we took. So, and then coming to a second question. So, and also, so there I want to say more. Yes, I know in ovarian cancer, I mean, you give, I don't know, chemo with some carbon compound. And I mean, that's what you typically do. And then you wait what happens, right? And then there might be relapse, etc. But for us, we are in a way agnostic to what is currently done, because the aim in our study was that the tumor board would also decide on off-label treatments. And in fact, of the 240 patients that we had, over 70% were off-label treated based on the data that we provided. And then it also depends on the patients that came in. And then typically it was not first-line treated patients, but it was very often those where actually the first, second, sometimes third-line standard therapies have failed. And the oncologists were like, all right, what are we going to do now? And then we got those cases. Because also ethically, I mean, that's where I think we can contribute, and oncologists start to do off-label treatments. So then, sorry, what was again the second question? It was
on the controls.
Yes, on the controls, exactly. So we did two things. One is that we compared. So we always calculated a reference that was the average of all the patients that we had analyzed. So then we could always say, this patient compared to the average is actually pretty high in PD-1 expressing cells or lower in PD-1 expressing cells. But this we did like as an internal reference, but then we also implemented external references, where that's the beauty of imaging mass cytometry. You can measure retrospectively patient cohorts, and based on those and even on clinical results that you might have, you can then determine what is low and high. And take this into the evaluation of what is the low and high expression.
Great, thank you, Anniina. I believe Jeroen from Cancer.io had also one question.
Yes. So congratulations, Bernd, on this fantastic technology, and I really admire the Swiss system that you set up. We've actually in the Journal Club presented, there was a Cell paper or some high-profile paper on it. And I envision this is what the future looks like. This is what it should look like. However, but I'm guessing, as you I think alluded to, this is kind of the treatment here is off-label, and your method is obviously not clinically validated, right? So this is kind of there is no alternative anymore, and therefore, in this case, for example, we do a combination that maybe in that tumor type does not exist. Are you planning to do a official, or is this maybe part of an official clinical trial? Because could you show that your method outperforms somehow others?
So yeah, you're asking obviously a key question. Are we better than current pathology? So far, this was an observational clinical study. So we did not have two arms where you would compare routine pathology versus this tumor profiler pathology. We did not do that yet, because to really go into such a very controlled clinical study, the technologies would have to be further developed to have certain certifications and the lab certain accreditations to actually do this. Because then if you go into such a trial, whoever comes in is randomly treated or not treated. And so then you have to fulfill many more guidelines than what we had to do now. Because in Switzerland, there's something called an individual healing attempt, where you are allowed ethically, but you always have to run it by the ethics board to give this off-label treatments in cases where otherwise there's basically no hope. But that's not a good setup for a controlled clinical study to actually prove that there's an improvement or not. But working towards that, because the tumor profiler is also very closely linked to the Comprehensive Cancer Center of Zurich. And in this setting, in combination, we try to come to the point that we can do controlled clinical studies.
Okay, so that sounds really good. And the other thing I was thinking is that because you're now doing this within the Swiss system, but are you thinking about offering this as a service in a commercial setting, where to private hospitals and such, where patients are at the end of their treatment and there is nothing more to be given within the standard treatment, and then often the oncologist has just no idea what to do. But your system, and then there are other methods on the market, or but this could be really an option for those patients all over the world, because you don't have any issue, right? You can ship this stuff very easily,
exactly.
And so
yes.
you are you get the results very fast. So technically, this would be a golden opportunity, I see, as to commercialize this. But I don't know if there's any ideas towards that.
No, I think there are very solid ideas towards that direction. Because, ultimately, if you want to bring something to patients, especially in an environment where there are very high regulatory boundaries and hurdles, and if you want to do this, for example, Europe-wide, you can only do this via a commercial entity. You cannot do this as a research lab or as a university.
Yeah, you would need to commercialize you need to set
exactly.
up a separate company to do that. No, absolutely. Heidi, are there any more questions? Because I would have a scientific question.
Yes, we received a couple of other questions.
Okay, perfect. Then I will.
And we have a couple of minutes too. So.
Yeah, then I'll go out and let the others get their questions.
Excellent. So there's one more. So great talk. Can you use this imaging technique to study drug resistance, say comparing before and after treatment imaging profiles?
Yes, absolutely. So we can compare with the antibodies that we use, how a tumor changed during treatment or when it became resistant. But so that's also an important point I want to stress. We measure 40 markers, and depending on your question, you would like or you want to actually adapt the antibody panel. So if you have a question of treatment resistance, then you probably want to include antibodies that might be good readouts for potential mechanisms of resistance. So, and then you could look at it more mechanistically, or you can do it on the level of just a phenotype. If you just want to see which tumor cell phenotype is the one that ends up being resistant or is more resistant than others. Of course, the tumor microenvironment gets a more and more important role in many of the resistant mechanisms. You can also focus on that. So yes, you can do it. That's a simple answer. But if you do it, you really should ask yourself what exactly you want to learn from this experiment, and then choose the panel accordingly.
That's a good point. Also, maybe good to address the tumor heterogeneity with this, like before and after. Like, what are the clones that would be?
Yes, absolutely.
For example. Okay, then I think it's time. Thank you, Bernd. And then we move on actually to the panel discussion. So I think all the panelists are here, and if you can turn on your videos and microphones, that would be great. And actually for the panel, we will have the speakers that you listened to earlier, but then also we'll be joined by Eevi Kaasinen, who is a bioinformatician and development manager in Helsinki University Hospital. And she's been working with this kind of similar problems and questions in translating complex bioinformatics systems into the hospital world. So she's also bringing some of the hospital perspective into the discussion. And then Miriam, if you could share the image, the third slide. Yeah, so this is just to kind of get us into the mood of the discussion. So as we just recently heard from the great talks, we're really now entering the era of complexity, and we could either say multi-omics. So this image is actually from Nature, like a technology review from this July, and it's an interesting read. So I pointed it out here if you want to go and read it. But it kind of highlights how the past decade we have witnessed an explosion in these single cell technologies like genomics and probably most prevalently the single cell RNA-seq. But this also means that we're starting to look at the tumors in such a detailed resolution like never before. And we're starting to generate these huge data sets and even massive layers of data that is kind of in this image. So that means gene expression data, chromatin accessibility data, RNA expression data, and as seen in many of the talks today, the protein level data. And it's really getting challenging from the computational view and software development view, and simply put it, how to just make sense of all this resulting data, and how can we eventually take all this and use it in the clinical setting, which was actually nicely addressed today in the keynote talk. But so first, for the panel, I have a general question. So this is for all the panelists, and everybody gets to speak. I can give you turns, but maybe we can start from Bernd's views. So now, since we're moving into this single cell and even spatial methods, what are the biggest advantages in investigating tumors in such detail? And I know you pointed this out already a little bit in your talk, but also maybe if I want to challenge you a little bit, then is there a chance that we're now getting too zoomed in and we cannot see the forest from the trees anymore at some point? But kind of what's your take on this?
So I think the great advantage is what I pointed towards initially, is that we actually do see now, even on different types of molecules, if you talk multimodal, it could be lipids, could be nucleic acids, can be proteins, that we have the chance to see what in tumors is deregulated, and based on that much better understand tumor biology, which always is the basis for better treatments, but then also identify features for patient stratification and or make predictions for patient treatment. So I would say the more data we can generate and the more types or modes of different types of molecules, the better it is in the end. But you're right. If we then look at the statistics, we start to run into huge issues. If we have thousands, tens of thousands, I don't know, I mentioned millions data points from one patient, and then we want to infer anything that's significant. I mean, you all know the issues. And I think here the art is on how to summarize the data. And that's also why we always love the single cell level. It's a great way to summarize all these many data points into something that we in a way still understand. And then with this much reduced data space, we always start with our analyses. And I think the advantage of this data is that you can choose yourself the granularity that you want to look at the data. Because, you know, if you have only extremely granular data, you don't have the option to go into high res. And if you have high res, you can still go back and imitate still more simple measurements. And
so I guess the argument is zooming in and out from the tumor, right?
Yes, and having the luxury of being able to zoom in, I do want to have rather than not having this ability.
Yeah.
Anniina, can I comment on, like, as a kind of a follow-up question? Is that report you are producing, or I assume you are having some kind of a multidisciplinary meeting tumor board in the study, and is the report you are generating, is it dynamic? So can you, or is it like a fixed PDF or something?
No, so in this tumor profiler project, we have a website where all of the data goes that has been processed, not the raw data. It goes also on the same server, but it's never visible. And then, like, all the scientific analyses out of the labs go onto a website. And then we have this pre-tumor board. The scientists and clinicians sit together and go through all of this data. And over time, we also learned which data are most interesting to clinicians and how to present them to clinicians. And there was also learning on the clinician side, because by now they see certain plots and like T-SNE plot or UMAP plot, and they immediately say, oh yeah, of course, I see this and that. That was also a really good learning experience. But to be honest, it's only a very small fraction of the data that's actually used from the clinician side. Because, you know, I mean, you know extremely well, they have like their 2 handfuls, 3 handful of markers they always want to know about. And ultimately, they just look at the data that somehow relates to those markers. And I would say 99% of the data we generate is never looked at by the clinicians.
Yeah, that's my point as well, like my experience. So for example, I mean, I work in ovarian cancer, and that's something that I think we still don't know enough about the immunology or the potential biomarkers or the treatments. So I think some of the other tumor types might be more, you know, amenable for this approach, like melanoma, where there's indications for anti-PD-1 therapy and so on. But in ovarian cancer, I would really, even with all that data, I would struggle to say this patient has a high PD-L1 and this patient has low or something. It's really difficult. Yeah.
I totally agree, but our hope is with this study to generate so much data that is not directly clinically useful that we can learn and make suggestions then what might be good markers to look at.
And in this sense, the multidisciplinary approach and, you know, you actually and sitting with the clinician is really critical, because it's the bridge can be huge between the research and the clinics.
I agree, and I have to say, we started this whole idea 2016, and it took quite a bit. I mean, now it's amazing how now the scientists here and the clinicians work with each other and exchange ideas, projects, and data. But it took quite a while until this grew to the point that it became so normal.
Yeah, and it really requires a lot of good communication, because the languages that bioinformaticians and clinicians speak is probably pretty different. So that's a good learning lesson for all of us, I think, from that side as well. Maybe a little bit siding to that or going to that direction, I could ask maybe Anniina and Eevi, who have been like working with this kind of problems. So kind of how to put these complex and detailed things for the clinical reality. So what is your view, and how do we take these processes and streamline them so that they can actually be used in the clinical setting? And especially maybe for Eevi, that what have been the challenges so far in doing such a thing? Of course, everybody can comment if you have some good points for this.
Well, from my point of view, I'm a clinician scientist, and I work half time in the clinic, treating patients with ovarian cancer or gynecological cancers, and then half of my work is in the research, and I do this multiplex IF analysis. And I feel that there's, even if I'm the same person, the clinician and the scientist is in the same person, the gap is huge. So from the patients, when I'm treating the patients, I could think of so many options and different therapies and biomarkers and tests that I could do, but then the drugs are not available. So that doesn't make any sense for me to order any tests, because even if the test is positive, that I will not get the drug for the patient. So I feel that there is this, as I know, this Cancer IO is really also pushing these, you know, the legislations and the fundings to get access to the drugs. So from my point of view, I mean, we are working on the research side heavily, developing biomarkers and developing understanding on the tumor microenvironment. But from the clinical side, it's still the biggest challenge is getting access to the drugs and who pays for them.
Yeah, exactly. So the whole clinical trial network has to support these kind of technologies, and the uptake of the drugs is really important part of that.
Just to add one little thing, and I also think in Europe, most of Europe, there are not enough MD PhDs because the educations are very much still the scientists and the clinicians, but MD PhDs that know both sides really well. They're unfortunately not so many, and I would hope and think in the future, given that we are all wanting to make precision medicine a reality, we need many more of this MD PhDs that bring both together in one person. Even then, I hear now that even then sometimes they can't talk to each other.
Yeah, and that comes heavily also from the political level. So we have to make sure that there's enough of these kind of like funding and positions for the doctors to be able to either part-time do research or then generate some kind of positions that you're doing something like Anniina is doing, because otherwise doctors have to do that in their free time, and that's probably not really rewarding for anybody. But maybe now I give the floor to Eevi to tell a little bit about the experience trying to set up these kind of bioinfo algorithms in hospital world.
Yes, thanks. And hello, everyone. So I work 50% in the clinical genetics laboratory and then 50% in the research. And I agree with Anniina that the gap is really huge. So my tasks are completely different in the different settings, and like what I mainly work on the clinical side is like validations, like we need to generalize all the methods to different sample types. It needs to be really automated, everything, and really like get everything working so that it's like kind of we need to solve the problems very fast because it's like a patient diagnostics. So it's very like also to open a new diagnostic like method, we need to consider a lot of different aspects before we want to open it, because we need to then know that whatever problem there is, we need to be able to solve it very fast. So I think that's kind of, I think it takes quite a long time to open a new analysis from the lab, because we need to consider so many things.
Yeah, absolutely. What was, Bernd, your experience from this working with the clinicians? And you said that you actually had to sit down and talk about the solutions and softwares, I guess. So do you share the experience with Eevi that it has to be really streamlined, or what was your experience from this? Yeah, sorry, Bernd, I was asking you, what was your experience working with the clinicians and trying to kind of get these algorithms and the system, the diagnostic system in use? Was it similar to Eevi's?
Oh, sorry, there was an issue. So, I mean, it's not in the routine use yet. I have to stress this, but it's within this observational clinical study. And when we started, we brought together clinicians that are really interested to work with basic scientists and to learn about this data and what it can do. And the other way around, the basic scientists, there are several labs involved, they were all really eager to assess what we are actually doing in the clinics. And so I think this was this upfront goodwill on both sides that really made it work, and this interest to ultimately figure out whether we together can help patients with what we do. And I mean, we are lucky that the clinicians that we are working with, they're very interested in science. Some have also MD, PhD background, and that also helped. But it was a long like hike together. Sometimes it was very tedious on both sides. But I think more and more we valued what the other side can do. Because sometimes I think basic scientists are a bit naive. They think, oh, we have this biomarker, now we can help the patient. And it was good for us to learn the clinical reality. And I think it was also in many aspects a good learning experience for the clinicians. And now at the point in Zurich that on both sides, there's so much excitement of what we have done, that we're really trying to make this a long-term structure in Zurich and Switzerland, where we always can work like together, and we have some running trials where we can then come in with the basic research results, etc. So in a moment, there's a lot of things are ongoing to solidify this collaboration and to really enable this as a long-term organization even.
And probably like once you have done it once and you have all those kind of pieces together, then it's probably much easier to do something or or tweak it a little bit, the existing setting or something like this, because then you already did it once and you know how it's done.
I mean, we're at the point that we know really how to do it. Now the issue is more to find the funding for something like that, because having such having a continuously running cohort and this many technologies, that's pretty expensive. And so that's where we are currently working at.
Maybe more for the technology side, I would want to ask a question from Päivi from SCellex. So I think you mentioned it a little bit already, but then we talk about single cell technologies, but we also talk about the spatial, the role of where every single cell is sitting in the tissue. So why do you think it's really important? And especially maybe for, if you can comment something, since it's an IO themed meeting, that why is it especially important to know where every single cell is sitting in the tissue, and for example, where the immune cells are and what type of immune cells they are? So maybe SCellex comment on this.
Yeah, of course, from the it's all about the same thing in the spatial, independent what is the method, is it antibody based or the sequencing based? But of course it's important to know, for example, look at the distance of the immune cells from the tumor cells, look at the phenotypes of the immune cells within the tumor. Where do they locate? Are they can they infiltrate to the tumor? And also, are they suppressed by the tumor cells, or are they cytotoxic? Can they do their job? Can we find better markers for hot and cold tumors in immuno-oncology terms, and so on? So I think the cell-to-cell interactions and where do they locate is of course important, and a very fascinating novel things that we can really study with the spatial technologies, whether it's antibody based or sequencing based. I really admire these antibody based technologies in such a sense that they are really quick and they can really and cost efficient, because in the sequencing based methods which we are working, the Illumina sequencing is the bottleneck, because I mean you can't really sequence million cells for all the transcript, because you run out of your grants already with the first sample. But with the immunostainings and the probe-based methods, where of course is you can really look at the in the high throughput way and the quick way. So, but thinking of this, I think of course these are complementing methods to each other, so that with the sequencing method you can find novel biomarkers. If you look at the old transcript, you might have then new candidates to create antibodies and then put them to the panel with the antibody-based detection methods. So if it's and in this sense, I would actually raise the question to Bernd, that how often do you update your antibody panel based on some novel findings in sequencing based methods? So are you basing your panels, I mean, with the data coming from the RNA-seq field, and how often do you need to add there more better markers based on the literature?
Actually, I think I have two answers to that. So first, in the newer projects in my lab, we indeed now do single-cell RNA-seq or spatial transcriptomics first on a small number of samples to actually get this comprehensive view on which markers are there, which pathways are present, etc. And we do this, as you say, to inform our panel design and to figure out which antibodies we should use. So I think that's one aspect. Another aspect where I think the genomics / transcriptomics and proteomic measurements are really complementary is, again, when it's about patient treatment, especially for off-label treatments. Because our experience here in Zurich is that in Switzerland, if you do off-label treatments, the oncologists are also can be made legally responsible. And so what you realized is that the oncologists were excited to see really independent or orthogonal technologies suggesting the same thing. Or if there was a big disagreement for them, it was like, okay, now let's understand why is there this big agreement. It helped them again to think about what they actually do. And And that's, I think, another layer that really showed to us that having this both types of data was very useful for the clinicians.
How about your marker panel? I mean, how useful it is? You think it's for the other cancers as well, or kind of is it kind of? Do you think that you can reach like general panel for many different cancers or cancer subtypes, or do you need to develop always a set of another panel for the other cancers?
So I think you need to develop for each tumor type its own panel. Yeah. Because you want to be able to distinguish the different tumor cell phenotypes. For this, you need unique markers. Then, as you all know, and don't have to tell you that there are different processes or pathways deregulate different tumor types. And these are all things you want to capture for a tumor type. And I don't know, if we might have one day, I don't know, 100 or 200 antibodies we can measure, then I think we can make a tumor agnostic panel, but with 40 now. And there's also, we have a separate immune panel and a separate tumor cell focused panel.
I think it's nice that it was mentioned that these technologies are really complementing each other, and that's where we actually enter the era of multi-omics that was now mentioned. Maybe one question, so how is it limiting factor that there's so much data? Like, have you ever run into, like any of you run into just that simply your analysis is too heavy, or that for basic research it's just not feasible to do something that the data would allow as such? Or is this an issue nowadays anymore, or not at all?
I can perhaps comment a little bit. I think in the beginning, I was as I was starting to do this multiplex analysis, I was kind of, if you do a basic run in Histocat and you look at all the spatial interactions and the heat map, you're like, oh, wow, like what? How can you differentiate which of these interactions are actually relevant? And so that kind of quickly pointed us to a direction where you need to have a question, you need to have some kind of a hypothesis by which you base your panel on, first of all, and then you ask the question from the data. So I think it comes to the same as in other technologies, that I cannot let the data speak to me. I will ask the questions from the data in a way. So because if you just start looking and exploring the data, it's really, it's huge. But on the other hand, there are some things that you need to be kind of alert for. Like, for example, Bernd showed very nicely where they noticed these, big spatial differences between the samples. And then you kind of start thinking about how could we model these computationally. And that's something that I always, try to keep in mind, and also tell people in my lab that when you're looking at these images, and if you see something very interesting or peculiar, keep looking at the images and look at the data if they might reveal something to you. So
yeah, and science is all about surprises. So that's a really good point also, to not be blinded by a question in a way. In my last minute, I just want to ask a little bit of tips for a really basic researcher. So what does this mean now for a person who doesn't know a lot of bioinformatics or R? Do we all have to become coders, or do you think that the softwares will catch up and we'll be fine, and we just have to learn a couple of new tricks? Or what do you think is the future development for the software side?
Well, I can comment that from the spatial transcriptomics side, the 10x Visium, which is now in the market. I mean, they have quite easy to use tool for the basic tools to visualize the data and look at that. But I think to really dig into the biology and to have a deeper look, I mean, people are also using command line tools, which are also now coming quite a lot of them in the literature from the academia side. So certainly just to have a quick look, I mean, these visualizing tools for the spatial data is good, but I think there are lots of things to do in the AI with the AI tools, specific tools, and that's what we are also aiming. If you find 40 markers spatial analysis complex, I mean, when you think we have 10,000 layers in Z layers for the for the data. So it really needs a kind of a machine learning to help.
Eevi, did you have some final words to close?
Yeah, I can comment from the clinical side that usually the tools we use, they need to be commercially available. So kind of they need to be very easy to use, and we need to have like support for them, because then as a bioinformatician, we cannot handle all the different methods and their tools and their code. So we need to also have like someone else to take the responsibility of the code. So then most of the tools we use are then commercial, or then they are very like golden standard pipelines that we implement then here.
Absolutely. Okay, I think we're running out of time. I could have asked so much more questions about this, but I'm happy that we were able to get such a vibrant discussion, and at least I really enjoyed this, so I hope you also did. And I want to thank all the speakers for the great talks, and Eevi for joining to the panel. And the recording from this webinar will be available in our website in a couple of days if you missed some part. And if you have any questions, you can always go to our website, www.cancerio.org, and find our contact information and ask something that was your question. And so we're closing with this, and thank you everybody, and have a good day.